how are these proteins get in there? How does a protein get through the two membranes, right? It has a single peptide, or it doesn't. Turns out they do. And the single peptides are slightly different. They're usually longer. They can be as long as 60 amino acids long. One thing that is true of all of these is that the single peptides on both mitochondria and chloroplasts have the structure that lets them form or makes them form an alpha helix, right? Where the residues on one side of that helix are all the same. Remember we talked about that before. So this is an amphipathic helix. And if you look at the red, you see the orange, the whites, the orange, the whites. The reds are all charged residues. And on the other side, the yellow that runs up the other side are Ru, Al, Pro, Phi. They're all isoleucine. They're hydrophobic. So essentially what happens is that the signal peptide on the proteins that go into mitochondria and chloroplasts, on their amino terminal end, there's the amino terminal methionine, form a helix that's amphipathic, and one side of that helix is hydrophobic. And that part of that helix, right, is recognized at the surface of the mitochondria. We're not going to talk about that receptor or anything. I do want to show you that, first of all, we do know that where proteins move into these organelles, two things happen. One, you notice that this protein here is moving, we show this protein is going to move in, right? And there's no ribosome there. So movement into organelles is slightly different than into the ER. ER is an organelle, but into chloroplast and mitose is different because it's rather than being co-translational, as we said for the ER, it's post-translational. So the proteins are totally synthesized, and after they're synthesized, they have a single peptide, and that gives them access to that mitochondria. Okay? Sort of interesting, right? I told you before that if the protein is made in the cytosol, it would fold somewhat. They couldn't get in. Well, it turns out that these proteins here, like this, have to have chaperones to keep them sort of in an unopened, molten kind of a form where they still can be threaded through. And one thing that does that with these proteins is HSP70. We talked about HSP70. So these nascent proteins, the mitochondrial and chloroplast proteins, are bound with HSP70, and it keeps them from, in that case, it keeps them from folding until they can be translocated in. Second thing this cartoon shows is that when proteins move in, the two parts of the membrane do come close to each other. They become opposed, very closely opposed. I don't know if I have one here. I guess I didn't bring one. But in an electron micrograph, you can actually see that where these guys are, the membranes are very close together. So the proteins move through at that site. So the first thing is that they're post-translational. Second thing, the membrane comes together. And the third thing is the process requires energy. And you can show that simply by temperature. If you change temperature in cells, at 5%, cells don't make ATPs, and they can't hydrolyze it. So if you grow cells at 5 degrees centigrade, they're not hydrolyzing ATPs. If you take proteins and you put them in there, like this guy here, what happens is that protein begins. Actually, it wouldn't begin. I guess what they've done is they've let it begin to translocate, switched it to 5 degrees, which kind of sort of stops us. It's transfer interruptus here. And then what happens is if you do that situation at 5 degrees, right, and you put a protease in, the protease hooks you up the protein because the protein's on the outside of the organelle. Whatever's inside is protected from protease, but on the outside, the protease on the outside, it's not protected and it chews it up. If you take this now and you increase it to 25 degrees, what happens is now the translocation continues because the energy is available, and so the protein is translocated into the membrane. So it's into the matrix of the mitochondria. If you then add a protease, the protein is protected. You can show that it's protected just by looking at a gel or using antibodies. Right? If you take this guy here and you treat it with a non-ionic detergent, that with the non-ionic detergent with small holes in the membranes, right? And then if you put in a protease, the protease can get through the small holes in the membrane and will chew up and digest your protein. So that shows that yes, translocation does occur, the proteins do get in, and yes, it does require energy. Right? If you don't have energy, it stops at 5 degrees, and the protein doesn't get in. There's a fourth thing that this doesn't show, and that is the translocation cells have a membrane potential. The inside, if you look at a cell membrane, a plasma membrane, the inside is negative with respect to the outside. Differs by about 70 millivolts. Mitochondria are even bigger. The difference is about 200 millivolts. So there's a membrane potential of about 200 millivolts across that membrane. If you dissipate that membrane potential, then translocation stops. So you need ATP, you need a membrane potential, you need, in the process, these two membranes come together, and it all happens post-translational rather than co-translational. Okay. Translocation occurs through translocons again, and there are two translocons that are needed. Remember, these membranes come together, and these translocons are called Tim and Tom. And Tom is the translocator of the outer membrane, and Tim is the translocator of the inner membrane. So translocation begins. Now this one here, this is a little more sophisticated model. It shows you that the receptor that actually recognizes the signal is actually sort of part of that translocon complex. It's attached to it. So the protein will bind to that receptor, and then from there get put into the translocon. But what happens is that when these guys are translocated, this guy here comes very close together with that, right? So this translocon essentially butts up against that. So the protein that moves into the matrix moves through Tom and Tim sort of in one step. It just goes right through that in that space, intermembranal space. One other thing that I didn't tell you, and I'll just tell you briefly, I think rather than starting on nucleus, I'll tell you. And that is, if you look at these membranes, remember I told you that some of these can be 60 long. And the reason they are is because there's more than one signal in the sequence. Proteins that end up inside, many proteins that end up inside this intermembranal space here, first are translocated through Tim and Tom, so they have a single peptide that gets them into the matrix. And then there's a second single peptide that transfers them back out through this membrane, right? Or in some cases, there's a double signal sequence that follows the Tim and Tom sequence that gets them in the membrane and anchors them. So there's, as they move in, that first signal is a part of the signal sequence that gets them through Tim and Tom is cut off by a single protease, right? It's cleaved away. And then there's a second signal, a start transfer and an anchor, that get them transferred and get them bound into the membrane. You can think of a protein that might go through like that and end up out here. What protein ends up out here? What's the only protein that you probably know that's in the intermembranal space? I'm going to tell Dr. Adams to put it on the file. ATP synthase is in the membrane, embedded in the membrane, yes, and that's one of them. It gets through like that. Although that's, no. I'll tell you why in a minute. Cytochrome C, you're in the right direction, right? But cytochrome C, that electron carrier that goes between complex, 3 and 4, is in that intermembranal space, and it does that. It gets synthesized into the matrix and comes out. ATPase, why not ATPase? Does anybody know that answer?
It's embedded in the intermembrane. Well, it is
It's in the intermembrane, but what else? So, who knows about the genetics of mitochondria? They have a chromosome, right? So when they replicate the chromosomes. In eukaryotic chromosomes, how many genes are there in our chromosome? How many proteins do we code for? Not very many. 9 or 10. What are the proteins that are coded for? ATPase and cytochrome oxidase. So apparently, those two membrane proteins are difficult to get to synthesize on the outside, put in the prosthetic groups and everything, and get it into that thing. So they're the only genes. How many genes did mitochondria originally code for? Where did mitochondria come from? Bacteria. So the original mitochondria probably had a minimum. I think the smallest bacterial genome we know is like 520 genes. So they had between 520 and E. coli, which is maybe 1,000 genes. But then over evolution, the vast majority of those genes have been put where? They got incorporated into the host genome. So they're now part of your DNA. Your DNA codes for all these proteins, the ones that are synthesized and get through. But there's still about a dozen or so, and it varies in organisms. It can be up to 50 or so. But I don't think any organism does the ATPases, especially the parts of the ATPase that are in the membrane, the inner membrane, and the oxidases.